Hi students, today we are going to the previous topic in pharmaceutics, polymers. This session introduce what polymers are, how they are classified and why they matter in pharmaceutical sciences. The concept we discuss here will later connect directly to drug delivery system and dosage form design. So in previous classes we discussed about what are the polymers and what are the different types of polymers and what are the sources we discussed in part 1, and today we are going to discuss it in part 2 and the remaining. So the three methods we are going to utilizations of polymers. One is the diffusion method and degradation and swelling. In last class we discussed about the diffusion, and today we are going to remaining slides we are going to discuss it. And second one, degradation method. So this slide, what we are going to explain is the working of biodegradable microparticle in drug delivery. Initially, the drug is trapped inside a biodegradable polymer microparticle. At this stage, there is no immediate drug release. When the microparticle enters the body, it comes in contact with the body fluids and enzymes. Because of this, polymer matrix slowly starts degrading. As the polymer erodes, the drug present near the surface of the particle is released first. With further degradation, more drug is released in a controlled and sustained manner. Finally, the polymer breaks down into harmless fragments and the entire drug is released. So drug release here depends on polymer degradation, not sudden dissolution. So this system helps in prolonged drug action and better patient compliance. So that is a degradation method. And next one, uh, water penetration. That is the swelling method. In this method, it is a typical oral tablet or pill releases the drug in the body initially. The tablet contains a drug-loaded dry hydrogels. In the dry state, the drug remains trapped inside the polymer network. After swallowing the tablet, uh, it comes in contact with the water from gastrointestinal fluids. The water slowly enters the tablet as shown by the arrows. You have to see the water will be enter into the drug. As water penetrates, the hydrogel swells. Because of this swelling, the polymer network will be open. Once the network expands, the drug molecule diffuses out from the tablet into the surrounding fluid. This drug release continues as long as water keep entering and the hydrogel remains solid. So here the drug release occurs mainly by water absorption, swelling and diffusion. This is the basic mechanism of drug release in a conventional oral tablet. So here we have to understand very clearly, it will be absorbed and to make the drug release. So it is the common method of the conventional oral tablet. The next one, what are the biodegradation of polymers? Okay, there are two types, biodegradable and non-biodegradable polymers. So now we have to discuss the biodegradation of polymers, which explains why those delivery system release drug over time. Biodegradation refers to chemical changes in a polymer that alter its molecular weight or otherwise its solubility. In simple terms, the polymer chains themselves are broken down. This is different from bioerosion, which is a physical process. Here, the polymer device gradually loses mass or weight from its surface. The polymer erosion mainly occurs by two mechanisms. What are the two mechanisms means? One is the hydrolytic mechanism. So in this mechanism, the water molecules attack the polymer bonds, especially ester of amide bonds leading to chain cleavage. The second is the enzymatic mechanism. Here, the enzymes present in the body specifically catalyze the breakdown of polymer chains. Both these mechanisms finally lead to polymer degradation, which enables controlled release and the safe elimination of the polymer from the body. So that is the biodegradation of polymers. Then hydrolytic dehydration. So the hydrolytic dehydration, how polymer degradation actually proceeds inside the body, continuing from the previous concept. In the hydrolytic pathway, the water penetrates into the polymer matrix and cleaves the polymer backbone. As these bonds break, the polymer become weaker, fragmented, and more permeable, which further facilitates drug will be released. In contrast, the enzymatic pathway is more selective. Here, the enzyme present in biological tissues attack specific sites on the polymer chains, accelerating degradation at a targeted regions. Depending on the polymer type, the degradation may occur from the surface or throughout the bulk of the device. This directly control the rate and pattern of drug release seen in biodegradable system we discussed in earlier. So polymer chemistry decides whether the drug is released slowly, uniformly, or in stages, making the biodegradation a key design factor in controlled drug delivery system. Then the second one, the enzymatic mechanism. So enzymatic degradation enzyme act as a biological catalyst that speed up degradation reactions. Hydrolase enzyme like protease, esterase, and glycosidase catalyze polymer breakdown. In some polymers, the enzymes cannot penetrate deeply due to high cross-linking. In such cases, the degradation occurs mainly at the surface known as enzymatic surface erosions. Then, what are the properties and selection of polymers? So, the highlights why polymer selection is critical means to achieve proper mechanical strength and biological performance, polymer properties must be understood. The key properties include solubility, viscosity, crystallinity, dissolution, behavior, dissolution behavior, erosion, and biodegradation. So these factors directly influence drug release rate and dosage form performance. So here the property selection we have to uh, focus on solubility, viscosity, crystallinity, polymer dissolution and polymer erosion and biodegradation. Then next slide we discuss about the characterization of polymers. So the characterization of polymers, uh, differential scanning calorimeter measures the phase transition energies. Then gel permeation chromatography determines molecular weight distribution. The glass transition temperature indicates the temperature at which an amorphous polymer changes from rigid to flexible. These techniques helps predict polymer behavior in formulation. So these are two types of characterization of polymers. So then the next slide we discussed about the application of polymers in solid dosage form. So in solid dosage form means for example tablets and capsules. In tablets the polymers act as binders, disintegrating agents, film coating materials and entry coating materials. So you know what is the purpose we are going to using for this type of excipients in tablet preparation, you know very well. In capsule areas, gelatin, a natural polymer, is the primary shell material. Each polymer serve as specific functional role in solid dosage form. So then, application of oral drug delivery system. The controlled drug delivery in oral system, the polymers regulate drug release over extended periods. In contraceptive and hormone delivery, polymers act as a reservoir coatings for sustained release. In diabetic treatment, the polymers functions as barriers between insulin and the bloodstream, enabling controlled action. So then in suspension, polymers like acacia. This slide we discussed about the um, what are the polymers, liquid dosage forms. The polymers used in suspensions and emulsions. In suspension, polymers uh, act as a suspending agents to maintain uniform dispersion. In emulsions, the polymers function as emulsifying agents to stabilize oil in water systems. This selection depends on the pH, solubility, and concentrations. So then, uh, applications in drug delivery. This slide we discussed the polymer-based drug delivery application. First thing, the polymers are used in film coatings, matrices, microcapsules, microspheres, eroding systems, osmotic pumps, implants, membranes, and bezels. Each application exploits a specific polymer property to control drug release. Then what are the applications of polymers? So these applications, more generally, the polymers used for mask the unpleasant taste, modify the drug release, and stabilize the formulation. Polyanhydrides are used for surface erosion system. The hyaluronic acid used in ophthalmic controlled release. The natural polymers act as a thickening agents and protective colloids. So then, the polymers used in uterine systems, implants, and elasticity-based applications. Some polymer provide strength, transparency, or hydrophilicity. Certain drugs themselves are polymers such as insulin, heparin, dextran, and albumin. Then another uses also the chitosan, dextrans, polysaccharides, and PLGA, PEG systems are using for the targeting purpose. These are used in hydrogels, transdermal delivery, wound dressing areas, colon targeting, and drug targeting. So here we conclude by showing the vast role of polymers in modern pharmaceutical areas. So you can understand about the polymers, the role used in the novel drug delivery system. Here we discussed what are the different types of polymers and there are the natural, semi-synthetic and synthetic polymers in which basis sources it will be converted to the polymers. Then what is the different mechanism of polymers and what are the applications of polymers. So I think you can understand the both polymers used in novel drug delivery system from part 1 and part 2. So thank you very much for observing, for understanding this polymer.